noticed how life begins? So simple, yet so astonishing. From beneath the quiet soil, life silently prepares, waiting for the right moment to emerge into the world. Look at these flowers, beautiful and full of color, yet they all begin as tiny, almost invisible seeds. A seed that appears still, yet holds incredible potential within. But to grow, a seed needs three essential things: water, oxygen, and light. Water awakens the seed from its long sleep. Oxygen activates life within. Enzymes start working. Food rivers are broken down into energy. Without oxygen, none of that can happen. The seed may stay moist, but it will never truly live. And what about light? For some seeds, like mung beans, light is not the trigger for growth, it's the guide. The tiny sprout will seek the light, grow toward it, and learn to adjust itself. Without light, the plant may still grow, but it becomes tall, fragile, and loses its color. Light is not just illumination, it's the compass of growth. Germination is more than just a biological process. It's a story of opportunity, direction, and balance. Water awakens, oxygen moves, light guides. And when all three are present, life begins to grow. It takes root, sprout, and finally blooms, like the flower we see now. This is germination, the very beginning of everything. Hello everyone, let me introduce myself. My name is Rizqiyah Almuthohharah from Hasanuddin University. Now, I'm going to show you my biology experiment entitled The Effect of Visible Light and Oxygen on the Initial Germination of the Mung Bean Seeds or Vigna radiata. In this experiment, I used some simple tools readily available at home. This included 4 small, clear plastic cups as planting containers and clear cling wrap to cover the cups. I also prepared a lidded container to create a light-free environment for protein groups. The material needed for this experiment are 40 uniform, healthy mung bean seeds, clean cotton as a growing medium, and clean water to moisten the medium. I used natural sunlight as a light source. The initial step of the procedure began with preparing the tools and materials. Cotton was moistened with clean water and filled into each glass as a growing medium. 10 mung bean seeds were placed on top of each. Next, the conditions of each group were adjusted. Group A was placed in the bright room and left open, exposing it to light and air. Group B was also placed in the bright room, but the tops of the glasses were tightly covered with plastic wrap to prevent air from entering. Group C was placed in the dark room, such as a cardboard box, but left open to allow air access. Meanwhile, Group D was placed in the dark room and also tightly covered with plastic wrap. After all the groups were arranged, I observed the seed development after 22 hours. Parameters observed included the number of seeds that enlarged due to water absorption, the time the first radical emerged, and shoots grew. Our results were carefully recorded for analysis and comparison between treatment conditions. After 22 hours of observation, the observation table shows that both light and oxygen availability significantly influenced the germination process of mung bean seeds, particularly during the seed enlargement, radical emergence, and shoot growth stage. Group A, light open, showed the best result in terms of seed enlargement 9-10 and radical emergence 9-10, as well as shoot growth 3-10, characterized by bright green color and upward growth. Group B, light closed, also showed a relatively high germination rate, but the pale color and limited shoot growth indicate that, despite the availability of light, the lack of oxygen in the closed container inhibits optimal respiration. This demonstrates that oxygen is a crucial factor for metabolic activation and energy production during the germination phase. Interestingly, Group C, dark open, produced the most shoots, 5-10, despite their pale yellowish color. This indicates that dark conditions trigger etiolation, which is rapid shoot elongation in response to the absence of light. However, because the environment is open, the oxygen supply remains sufficient to support early metabolism. Meanwhile, Group D, dark closed, showed moderate result in all parameters, but still exhibited pale color. This confirms that darkness does stimulate initial shoot elongation, but the limited oxygen in the closed space still limits optimal cellular activity. Thus, it can be concluded that the light reaction is necessary for the formation of green color and directional shoot growth, while dark conditions accelerate initial elongation. However, oxygen is essential under raw conditions because it allows cellular respiration and provides energy during incubation and embryo activation during germination. To support this experiment, we refer to the result of search by Tangkapan et al. 2018 on Vigna radiata, or mung beans, which examined the effects of light, darkness, and salinity on the germination process. Study showed that mung bean seed germination was more optimal in the dark condition. In darkness, 100% of seeds germinated on the third day and grew to 18 cm by the sixth day. Conversely, under sunlight, only 70% of seed germinated with shorter sprout. This is because in the dark, carbon dioxide gas is undisturbed and oxygen remains available, supporting the seed's respiration process. Therefore, as stated in the journal, darkness has no effect on carbonic acid gas, and the oxygen remains undisturbed, favoring germination. Hence, it's more correct to state darkness as a germination factor and light as a hindering factor. In other words, darkness favors germination, while light is actually an inhibiting factor in the early stage of the seed growth. To support this experiment stating that oxygen is needed during the germination process, we refer to research published in Seed Science Research by Francois Carbonell in 2022. Oxygen is stated to be a key factor in seed germination, as it allows the resumption of respiration and the reactivation of metabolism as the seeds begin to absorb water, a phase called inhibation. When oxygen is available, seeds can undergo aerobic respiration, a chemical process that produces ATP and reducing power, which are energy and reducing molecules that are crucial for cellular activity. Without oxygen, this process is inhibited. Anaerobic respiration cannot produce enough energy to support optimal cellular activity, so the seeds fail to continue germination. This experiment aims to examine how light condition and oxygen exposure affect the initial growth process of the seedlings, especially the emergence of the radicle or the first root. This experiment showed that oxygen is a major factor supporting seed germination through aerobic respiration, which produces energy while light influences the direction and quality of seedling growth. The best results were obtained in bright, open conditions, but the highest number of shoots emerged in dark, open areas because dark reaction trigger etiolation. This finding aligns with research by Tangkapan et al. 2018, which stated that darkness actually accelerates germination and identified light as an inhibiting factor. Furthermore, research by Corbinio 2022 confirmed that oxygen is crucial in activating metabolism during imbibition, as without it, respiration and shoot growth are not optimal. That's all my experimental video. Thank you!